As the winter melts into springtime, an explosion of color heralds a new season. Angiosperms have been using flowers for reproduction for millions of years, but humans have turned these colorful ornaments into symbols of beauty and art. Ikebana, the Japanese art of flower arrangement, dates back into the 7th century. This spiritual art form is meant to bring the viewer closer to nature and merge the indoors with the outdoors. Paintings, poetry, and even ceramics that date back hundreds of years provide records of the ongoing fascination humans have with flowers. Before we get into the flower itself, let's talk about inflorescence. A plant's inflorescence describes how its flowers aggregate on a single stalk, called the scape or peduncle. The flowers usually sit atop a leaf-like bract. An inflorescence will not be interrupted by foliage leaves. If an inflorescence has individual flowers directly attached to the peduncle, it is considered sessile. Sessile inflorescences include the spike and head. Alternatively, an inflorescence may connect individual flowers to the peduncle by another stalk called a pedicel. The panicle, raceme, corymb, and simple and compound umbel inflorescences are examples of this type of inflorescence. There are also some unique inflorescences. The Catkin inflorescence has unisexual flowers that lack petals, and the spadix is a type of spicate inflorescence where the flowers are tightly packed onto a fleshy stem and surrounded by a large bract known as the spadix. As an inflorescence develops, the flowers at the base of the plant may begin maturing first, and a wave of maturation will sweep upwards towards the tip of the plant. This is called acropetal development. The inverse of this, basipetal development, occurs when the flowers at the tip of the plant mature first. Now, let's talk about the flowers themselves. Flowers have 4 principal organs arranged in distinct whorls: the calyx or sepals, the corolla or petals, the andrecium or male stamens, and the gynecium or female carpel. The sepals and petals together are called the perianth. If the sepals and petals can't be differentiated from one another, they are called tepals. Sometimes the perianth may display streaks or markings called nectar guides, which guide pollinators to nectar glands. The andrecium is made up of individual stamens composed of an anther and a filament. The anther will usually be lobed, and each lobe constitutes its own pollen sac. These pollen sacs have an outer layer that will open or dehisce when the anther reaches maturity, and an inner tapetum that will nourish the pollen grains while they develop. Within the tapetum, diploid mother cells divide by meiosis to produce a tetrad of haploid microspores. A single-celled microspore is called a pollen grain. During microgametogenesis, the microspore will divide by mitosis to produce a vegetative cell and 2 sperm cells. Some plants will release their pollen grains individually, while others, like orchids, will release all of the pollen grains in one anther sac as a collective pollinium that can join with other pollinia to form a pollinarium. We will discuss pollen structure more in the video on pollination. The gynecium consists of the flower's female carpel, which is made up of the stigma that receives pollen, a style, and an ovary, which holds the ovules. Angiosperms are distinct from gymnosperms because they have ovules encapsulated within an ovary. Ovaries can show diverse modes of ovule attachment, called placentation. In parietal placentation, there is only one chamber or locule within the ovary, and ovules attached to the ovary wall. In axial placentation, there is a central column of tissue that divides the ovary into multiple locules, and the ovules attached to this tissue. In free central placentation, the ovules still attach to a central column of tissue, but there is only one locule. Finally, in basal placentation, a single ovule attaches to the base of a unilocular ovary. We can use the many variations in floral structure to categorize flowers. Complete flowers have all 4 whorls, while incomplete flowers will lack 1 or more whorls. Perfect flowers will have both an andrecium and a gynecium, while imperfect flowers will lack one or the other. While a complete flower is necessarily perfect, a perfect flower is not necessarily complete, since it may lack petals or sepals. Some flowers, like daisies, have equidistant organ spacing in each whorl and show radial symmetry. In flowers like orchids, however, organ spacing and size are not equal, and thus they display bilateral symmetry. Monocot flowers will usually have whorls arranged in multiples of 3, while eudicot flowers have whorls arranged in multiples of 4 or 5. Flowers are made even more complex by floral fusion. In some flowers, organs of a single whorl, like the petals, may fuse together. This is called conation. Organs from different whorls, like stamens and petals, may also fuse. This is called adnation. Adnation of different whorls can result in different floral architectures, which are defined by the position of the ovary. In hypogeny, all of the floral organs sit beneath the gynecium, so the ovary is superior. In perigyny, the calyx, corolla, and andrecium adnate to form a tube called a hypanthium, which surrounds the ovary. This tube hides the ovary, but the ovary is still technically superior to the other whorls. In epigyny, however, the outer whorls will fuse above the ovary and bury it, resulting in an inferior ovary. Clearly, floral structure is incredibly diverse. While we may value floral varieties simply for their aesthetic beauty, this diversity has huge evolutionary significance. Different floral structures have enabled certain angiosperms to specifically attract pollinators or capitalize on wind pollination. So, the next time you buy flowers, think about the millions of years of selection that have produced that bouquet.